So these two are the daughter cells.
Then this would be the mother cell.
Right. And the chromosomes that are in the daughter cells should be identical to what's in the mother cell.
Which they are.
What are you two up to?
We're just using these models to demonstrate the process of mitosis, where one cell divides to produce two identical cells.
Okay. And Shailene, what are some of the characteristics of the daughter cells produced by this process?
Well, the chromosomes in the daughter cell are the same number and same kind as in the original mother cell.
In this case, the mother cell has 4 chromosomes, and the two identical daughter cells have 4 chromosomes also.
Good. So, Mark, I'd be correct in saying then that mitosis is a process which helps maintain chromosome number in a species.
Right.
Okay, let me give you a different situation now. In sexual reproduction, a sperm unites with an egg to produce a single-cell zygote. How can chromosome number in the species be maintained when 2 cells unite to form 1?
The sperm and egg would have to have fewer chromosomes than a normal body cell.
Yes, that's right. They'd have to have exactly half the genetic makeup, or 23 chromosomes.
So when they combined, the normal number of chromosomes would be maintained.
All right. Every cell in our body has 46 chromosomes, 23 pairs. That's called its diploid or 2N number. Now, the gametes have 23 chromosomes. They're called haploid or N.
So the human sperm and egg cell would have 23 chromosomes, or half the diploid or 2N number.
Right, but not just any 23 chromosomes. One of each of the 23 homologous pairs enter the gamete.
What does homologous mean?
Homologous chromosomes are a pair of chromosomes found in a diploid cell. Let me use the licorice here to demonstrate. One comes from the egg, one comes from the sperm. Early in meiosis, the two of them line up and then separate to form the gametes.
Meiosis?
Yeah, meiosis. It's called reduction division. It's the process in the body that leads to the formation of gametes. A single diploid cell divides to produce 4 haploid cells. Now, can you think of how this might happen?
If these licorice represent chromosomes, one cell could have 2 chromosomes, and when it divided, we'd have 1 chromosome in each cell.
No, that can't be right.
Why not?
Because we have to produce 4 cells, and we only have 2 here.
I remember in mitosis, chromosomes duplicate in this process. What happens if it happens here?
What if the cells divided again, and then we end up with 4 cells, each with 1 chromosome, or half the genetic material we started with?
That's very good.
The number of chromosomes vary among different species. Some ferns have more than 1,000 chromosomes in their diploid cells. Dogs have 78, goldfish have 94. Even an amoeba has 48 chromosomes. As far as biologists can determine, there is no apparent significance between chromosome number and species complexity.
Meiosis, there are 2 nuclear divisions, followed by division of the cytoplasm, cytokinesis. One of the differences between mitosis and meiosis is that meiosis has 2 distinct divisions, called meiosis 1 and meiosis 2.
But what about the division phases we learned about in mitosis?
They're present in meiosis as well. Now, do you remember?
Prophase, metaphase.
Anaphase and telophase.
Good. Meiosis, they're repeated. The interphase between meiosis 1 and meiosis 2 is usually very short or sometimes not even present. But prophase, metaphase, anaphase, and telophase are numbered 1 or 2, depending on whether they occur in meiosis 1 or meiosis 2.
Oocytes are produced in follicles within the ovaries. Interestingly, the ovaries of a female fetus already contain all the cells that will eventually mature and become egg cells when the female reaches puberty. Prior to meiosis, the chromosomes have replicated during the S or synthesis stage of interphase, just as they did in mitosis. When the chromosomes become visible at prophase 1, each one consists of 2 identical sister chromatids, joined by a centromere. The chromosomes now pair up with their homologous counterparts. Homologous chromosomes are pairs of chromosomes that carry genes controlling the same inherited traits at the same positions on the respective chromosomes. One member of each homologous pair is inherited from the mother, and the other comes from the father. These homologous pairs form a tetrad of 4 chromatids. During the pairing up of homologous chromosomes in prophase 1, a chromatid from one chromosome may overlap with a chromatid from its homologue. The point of contact is called the chiasma. It is at the chiasma that one segment of a chromatid is exchanged with a corresponding segment from the second chromatid. This process is called crossing over. Crossing over results in variations of genetic information in the gametes. By the end of prophase 1, the nuclear envelope has deteriorated and spindle fibers have appeared. At metaphase 1, the homologous pairs of chromosomes line up as pairs along the equatorial plane of the cell. The spindle fibers connect the centromeres of the homologues to opposite poles in the cell. During anaphase 1, the homologues separate from each other and are pulled to the opposite poles by spindle fibers. Note that the sister chromatids remain attached to the centromere and move as a single unit to the poles. By telophase 1, the homologues have arrived at their opposite poles. Each pole now has a haploid set of chromosomes, but each chromosome still has 2 chromatids. Cytokinesis usually begins forming 2 daughter cells. At this point, new nuclear envelopes may or may not be formed, and the daughter cells begin preparation for meiosis II. In meiosis II, if nuclear membranes were formed, they disappear, and the chromosomes again become visible. A new spindle apparatus appears at right angles to the one formed in meiosis I. As this stage proceeds to metaphase II, the chromosomes align the equatorial plane in single file with their sister chromatids, facing the opposite poles. The spindle fibers again attach to the centromeres. In anaphase II, the centromeres split and the sister chromatids separate and are pulled to the opposite poles. Chromatids are now considered chromosomes. With the onset of telophase II, the spindle fibers disappear and new nuclear membranes are generated around each set of chromosomes. As the nuclei reappear, cytokinesis divides the cytoplasm and 4 haploid daughter cells are formed. This process occurs in all vertebrates.